Bismillahirrahmanirrahim. This is Biochemistry 2 course, code Chem 342, and today we are having lecture 23. Our today's topic is active sites of enzymes. This is Dr. Naeem Khan, Associate Professor, Department of Chemistry, CUS. In today's topic, we will talk about the active sites of enzyme description. We will highlight about the active sites characteristics. What are the important properties of active sites are having? To have a start, active sites. Active sites is that part of enzyme which binds to the substrate for chemical reaction. We know that the substance to which an enzyme reacts is called substrate. So for a chemical reaction to catalyze, an enzyme is required to bind to its substrate. The point where an enzyme gets attached to the substrate is called active site. So the active site is that part of the enzyme by which the enzyme binds to the substrate, gets attached to the substrate for the chemical reaction to be catalyzed. So active site is the part that links the enzyme to its substrate for the catalyzation of chemical reaction. Active site contains groups called catalytic groups. The active site of enzymes contains some special groups, and these are the groups which are required for the catalysis. The catalytic groups means the groups required for the catalysis of the enzyme catalytic reactions. Enzyme catalyzed reaction, they are directly involved in the bond breaking and bond formation. We know that in a chemical reaction, some bonds are broken and new bonds are formed because a new substance is formed. So in a chemical reaction, bonds breaking and bond formation are involved. Catalytic groups are the groups present in the active sites of enzymes are those which are directly involved in the breaking and formation of bonds, and hence we can say that the catalytic groups which are present in the active site of enzyme, they are directly participating in the carrying out of chemical reactions. Okay, we know that enzyme is basically made up of two parts, most of the enzyme. Some enzymes are only proteins, but majority of the enzymes are those which are made up of proteinous part and non-proteinous part. And we have already studied in the last lecture that the proteinous part of the enzyme is apoenzyme. Here, we are saying that active site is the part of apoenzyme. Means active site is present in the proteinous part of the enzyme. It is not in the non-proteinous part. The basic functional part of the enzyme is apoenzyme. So we say that active site, they are the site required for the catalytic activities of the enzyme is present in the proteinous part or apoenzyme part. This is the scheme. The proteinous part of enzyme is called apoenzyme. When it is linked to the non-proteinous cofactor, this becomes whole enzyme, and this is the functional enzyme in majority of the cases. So active site is attached here with the apoenzyme part. Catalytic groups of the active sites are amino acids. Yes, if it is the part of apoenzyme, it means it will be made up of only amino acids. So thus we can say that active site is made up of amino acids. Amino acids in the active sites are brought together due to the folding and refolding of polypeptide chains of apoenzymes. We know that proteins have structure, maybe primary structure, secondary structure, tertiary structure, and maybe having quaternary structure too. In the tertiary structure, there is folding and refolding of the polypeptide chains. So we explain here that the amino acids in the active sites of enzyme, which are catalytic groups, they are brought together from different parts of the whole enzyme due to the folding and refolding of polypeptide chain. I mean, when the apoenzyme polypeptide chains are folded and refolded upon one another, so some groups are exposed and they are brought together in the active sites and they are then called catalytic groups. So the amino acids, which are the catalytic groups in the active sites of enzyme, they are brought together due to the folding and refolding of polypeptide chains of the apoenzyme part. Characteristics of active sites. First one is the nature of the active site. Active site of enzyme has dual nature. It's basically having double functions. So that's why it's of dual nature. First, active site is having catalytic site and it is having substrate binding site. So the active site is basically twofold. Two regions are there. One is that which is having groups that are required for the catalysis of chemical reaction. And the second is groups are there which are required for the attachment to the substrate. It can be shown like this. We know that in enzyme and a protein chain, we have two ends, nitrogen end or that end having free amino group and the other end will have the free carboxylic group. Because if the amino acid on one end of the polypeptide chain is having free amino group, so on the other hand, the carboxyl acid end of the amino acid will be exposed. If this is the active site of enzyme, so it is having dual nature because some groups are here in this part that is required for the substrate binding and some groups are here which are required for the catalysis of reaction. So active site of enzyme is having dual nature. It contains groups for substrate binding and it contains groups for the catalysis of chemical reaction. So active site is having dual nature. It contains group for the catalytic catalysis reaction and it contains groups for the binding to the substrate. Second is the shape and size of the active site. It must be noted that active site is very small compared to the total size of protein. And here it is mentioned that approximately active site is 10 to 20% of the enzyme size of the protein part. So it's a very small part and its shape may be in the form of crevice or cleft. Means crevice means it is in the form of a small crack like this where substrate will get entered and then interaction will taking place and the recatalysis will take place. Or it may be in the form of a large opening, a cleft. So a substrate will come close together and a chemical reaction will be catalyzed. So active site is a very small, up to 10 to 20% of the enzyme size, and its shape may be a crevice or a small crack, or it may be in the form of opening cleft. Nature of bonding. Bonding of the active site with the substrate may be electrostatic interaction, because we know that there are positive and negative charge groups. Amino acids are charge groups. There are negative groups such as acid groups, or there are several positive groups such as NH3 positive. So if a substrate charged oppositely charged come close together, there may be interaction of the opposite charge ends. So electrostatic interaction will develop. Also, hydrogen bonding is there, because we know that in the amino acid there are several um, electron rich and more electronegative groups such as COO negative of the acid group, or NH2 of the lysine group. And here, if the substrate comes together which is having electron rich species or is having electropositive hydrogen, so there will be interaction of the electron rich species lone pair and the electron deficient hydrogen, and this is called hydrogen bonding. Third is, sorry, this was the hydrophobic interaction. And in the hydrophobic interaction, the water loving groups comes and gets linking the substrate with the enzyme. The third is the hydrogen bonding, where the electron rich species of the lone pair of electron rich species with more electronegative group, and the electron deficient hydrogen of the other molecule gets attached, so hydrogen bonding will be there. And the fourth one is the van der Waals forces, where like dipole-dipole interaction or London forces come into play, because the amino acids have several non-polar groups, such as in case of alanine, there is methyl group. So due to the high speed of electron speed, they get distorted from their normal path, and that's why there is big attraction become developed due to the temporary dipole created. So the bonding of the active side with the substrate may be electrostatic due to different oppositely charged groups, and it may be because of the hydrophobic interaction with water loving groups comes into play between the two, between the substrate and active site, and they bind them together. Maybe hydrogen bonding because of the hydrogen electronegative species lone pair, and the electropositive hydrogen becomes interacted, and this may be because of the dipole-dipole interaction or the van der Waals forces. Active site composition. As we already mentioned, active sites are composed of amino acids only, because it is the part of apoenzyme of enzyme. This may be more than one chain of amino acids. It's not important that how many amino acids, depending upon the enzyme structure and enzyme function. They are containing chains of amino acid, and maybe more than one chain of amino acid brought together, exposed to the active site because of the folding and refolding of the enzyme. They are brought together by the tertiary folding of enzyme protein. We already explained that the protein structure folding and refolding results in the exposure and bringing together of the catalytic groups of amino acids at the active sites. Examples of exposed group. And at the active site of enzyme may be having SH group, such as of cysteine amino acid, and this SH group is required for the disulfide linkages. So whenever a substrate comes in contact to the enzyme, disulfide bond can be established. And disulfide linkage SH group of the cysteine at the active site of enzyme is exposed. Similarly, amino group may be there at the active site, such as that in case of lysine. So if lysine amino acid is exposed at the active sites, and this lysine in nitrogen is more electronegative, and also nitrogen is having lone pair of electron. So this can establish hydrogen bond. If NH2 group, amino group of lysine is exposed and the active site and substrate comes together, there is the possibility of establishing hydrogen bonding. Similarly, COO negative groups may be there because of the aspartic acid. Aspartic acid is an amino acid is having dicarboxylic groups, dicarboxylic acid groups. So if this one of the COO negative is exposed in the active site, it's a negative charge, it will be establishing the electrostatic bonding when a substrate come close to the enzyme active site. Fourth one is the methyl group. For example, the non-polar group of the alanine, if exposed at the active site, and if the substrate come close together, then there is a possibility of van der Waals forces may be established. So the active sites composed of amino acids, and amino acid chain may be more than one, and they are brought together by the tertiary folding of enzyme protein. This we know. The groups exposed may be having SH groups for the establishing of disulfide linkages. These may be having NH2 groups for the hydrogen bonding, may be having COO negative groups for the electrostatic bonding, or may be having non-polar groups such as methyl group for the van der Waals forces interaction. Substrate binding mechanism. There are two possible mechanisms which are already explained. One is the key and lock model, and this was explained by Emil Fischer in 1890. And according to this model, enzyme and substrate are just like lock and key. First, the enzyme and substrate come together, they become fixed into one another, just like the key fixed in the lock, and this result in the formation of enzyme substrate complex. And then the products are released and enzyme is released. Products are formed and enzyme is released. This is a key and lock model which says that enzymes and substrate are just like lock and key. They are forming enzyme substrate complex, and then the product is formed and enzyme is released. Second mechanism is induced fit model, and this was explained in 1958 by Donald E. Koshland. And according to this model, enzyme and substrate are not like lock and key. Actually, when enzyme and substrate come close together, there is a conformational change. Conformational change takes place, and then the enzyme become fit into the substrate. Because of the conformational change taking place, the enzyme becomes a key. The enzyme and substrate becomes key and lock, and then fixed together, forming enzyme substrate complex, and this then result the formation of product and enzyme is released. The difference between the two models is the Emil Fischer model says that enzyme and substrate are just like lock and key, but here the Kushland model says actually the lock and key model forms after the conformational change. In both the models, enzyme come in contact to the substrate and become enzyme substrate complex, and then products are formed, enzyme is released. Mechanism of catalytic sites, we explained that enzyme come in contact with the substrate, we get enzyme substrate complex, and then products are formed and enzyme is released. What is the mechanism? Mechanism is, this is the, if we show the reaction progress by a graph, this is the free energy change, and here is the reaction progress. When this is the substrate energy is this one, and when it becomes change to product, its energy becomes decreased. We know that the product energy is always less than the substrate, and this is the free energy change for the reaction. Now, in case of uncatalyzed reaction, the free energy change for the uncatalyzed reaction in the to reach the transition state is this one. Look, this is a big amount. And in case of catalyzed reaction, the free energy change required for the transition state formation is small one. So what enzyme is doing? Enzyme is actually decreasing the energy required. Yeah, you can say activation energy for the formation of enzyme substrate complex and in the products form. So actually when substrate gets attached to the enzyme, it bring about conformational changes, due to which the energy required for the transition state formation is decreased, and hence the product is formed easily. So in this way the rate of reaction is increased. Dear student, Western Todd book, GL Jain, both Western Todd and GL Jain books are recommended, and please have a thorough study in these books and um, develop a thorough concept of each point we discussed today. See you next time. Until then, Allah Hafiz.